Okay, so lecture 12, we're going to discuss the topic of northern blotting analysis or northern blot analysis. So, lecture 11, we talked about southern blot analysis. So, southern blot analysis allowed us to screen the existence of matching DNA strand. Okay, the matching is done between your probe sequence and the target DNA sequence. So there's a process called hybridization. Through hybridization, you are able to isolate sequence matching between your probe sequence and the target DNA. That's what southern blot hybridization was. Okay. Then how about northern blot hybridization? Sounds like directions, right? Southern and northern. The name came from the direction. So northern came from that direction difference as opposite. So, but the principle wise, it's very similar to the southern blot hybridization, but the main difference between southern blot and northern blot is your target molecules. So southern blot, you are hybridizing your probe against the DNA molecules, right? DNA molecules they were separated on a gel through electrophoresis, and then those DNA molecules were moved from the gel to the membrane, and you hybridize. And how about northern blot hybridization? Pretty much the same, but the only difference is your target molecules are RNA molecules here, not DNA. Okay, but you still have a probe DNA, single-stranded, that's labeled with something. Okay, that generates your signal, maybe radioactive isotope or fluoropore or chemiluminescence, whatever that is. Okay, so your single-strand DNA probe is still hybridizing to target molecule, but the difference is the target molecule in this case are RNA molecules. Okay, that's the only difference. So when you talk about RNA molecules, we gotta know how to deal with RNA molecules. Okay, so you need to be able to understand how you can isolate RNA molecules from the living cells, and then how you can purify them and how you can analyze them, those molecules. So especially RNA molecules from eukaryotic cells. Okay, so if you look at this diagram, so the first question, what are RNA molecules? Okay, how are they made? Okay, why are they important? Okay, we know that RNA molecules are made from a cellular process called transcription, right? So if we are dealing with a eukaryotic cell, yes, the transcription happens inside of a nucleus. Okay, from DNA, okay, your RNA copies are being made. So RNA polymerase will transcribe your RNA molecules. So this yellow strand, in this case, is RNA molecules that's being transcribed from your DNA. Okay, so that's a transcription. Then you make RNA copies. Okay, once RNA copy is made, it'll be transported from nucleus to outside. Before that, there's a process called splicing. This explains kind of splicing process. In eukaryotic cell, there are so-called introns between the coding region axons. So there are axons that are flanked by introns. Introns should be removed from your RNA before it's transported out. So it's intron removal splicing process that's been explained. So when your introns are removed and exons are connected, your RNA is transported to outside where the translation will take place. Translation is taking place where your ribosome is located, most like ER and the plasma reticulum, membrane surface. Then translation will take place. You'll make your proteins. So RNA is used for that purpose. But besides that, there are so many different RNA molecules that will be never translated. Okay, those RNA may exist within your cell that will serve different functions. For example, this ribosome structure is actually made with ribosomal RNA and ribosomal proteins. Okay, there are so many other RNA molecules that act like enzymes. We call them ribozymes, and there are so many RNA molecules that are involved in even transport of this RNA from inside of nucleus to outside. Many of them are made with RNA. As well as this molecule right here that's involved in RNA splicing, that's also made with a bunch of RNA molecules. So actually RNA molecules serve many important cellular functions. Okay, so typically mammalian cells has about 10 to 5 microgram of RNA molecules in them. One single mammalian cell, so a lot of RNA. And what are the RNA molecules that exist inside of a cell? What do they do? About 80 to 85% of ribosomal RNA, okay, and it's taking up most of RNA molecules. So ribosome, there are a lot of those, and ribosomes are made of ribosomal RNA. abundant quantity. Rest of RNA molecules contain transfer RNAs. Remember transfer RNA, that's involved in the translation process. tRNA molecules will carry amino acid, and then it's going to recognize the codon on your mRNA because it's got an anti-matching codon. Okay, and the snRNA, that's the RNA involved in splicing. Okay, so there are a bunch of different RNA molecules that are serving important functions. And then very small amount of RNA will serve the function of messenger RNA. Only 1 to 5% of the total RNA belongs into this group. Okay, so there are so many RNA molecules that are serving important functions. Okay, and then the difference between RNA and DNA. As we know, RNA is made up with ribose sugar. Okay, so ribose sugar has OH, two OHs on the ribose. Okay, and then RNA has U nucleotide instead of T, thymine-nucleotide, uracil. They have another important thing. RNA exists as single-stranded conformation. DNA exists as double-stranded conformation. Right? There are differences. Okay, so how would you isolate RNA molecules from cells? So RNA, DNA, the difference is minor, although there are three distinctive differences. But compared to RNA against protein, RNA is very similar to DNA. So if we can isolate RNA and DNA from cells, you can also isolate RNA molecules because they are very similar. Once you isolate a bunch of cellular RNA molecules, then your question is, can you fish out mRNA molecules only from other RNA molecules? If I go back, I told you mRNA only occupies about 1 to 5% of the total RNA population. So it's very minor. So there are a whole bunch of RNA molecules that are not messenger RNA. So if you want to do the hybridization process, node block hybridization process, it targets the mRNA molecules, not other RNA molecules. That's why this question is kind of relevant there. Can you isolate mRNA molecule only? Okay, it turns out the answer is yes. Especially in eukaryotic cells, eukaryotic RNA molecules, mRNA especially, when they are processed, meaning when their introns are removed out, not only their introns are removed and exons are connected, also there's another modification. Okay, another modification is their 3' end tail has a long poly-A's. Okay, polyadenylation will take place, so they'll have a long poly-A tail there, and the 5' end will be methylated. That's 5' end methylation. Okay, those are two main, modification of your mRNA molecules. So if you think about that, they have a long A tail. You can use that feature when you try to fish out mRNA molecules because they have long A tails. Okay, so if you have a column chromatography that has poly-T nucleotide, T on them, then it'll bind with your mRNA that has poly-A's, AAAs. So A2 match will take place. By using that, you can fish out mRNA molecules. So yes, indeed, you can isolate mRNA molecules efficiently if you use that column. Another issue in dealing with RNA molecules, okay, RNA molecules are very susceptible to the degradation. So when I say degradation, that's different from denaturation. Degradation is what? It's an enzymatic process. There's an enzyme involved, and normally those enzymes will cut up nucleic acid into nucleotides. What I mean, those enzymes will break down those phosphodiester bonds. Those are covalent bonds. Okay, so if you look at this RNA molecule, between RNA nucleotides, there are covalent bonds. Those are phosphodiester bonds. So there are enzymes that can cleave up those phosphodiester bonds. Okay, so that's a degradation process. It turns out there are so many of those RNase enzymes, ribonucleotide enzymes, that can cut up RNA molecules. They're almost everywhere. That's the evolutionary, benefit that the living cells have, because there are so many viruses that are made up with RNA genome. Okay, so we have a defensive mechanism in our cells, so on our skins, against those RNA viruses like coronavirus, for example. They have RNA genome. So before they come and insert their genome to infect us, our enzymes that can cut up those RNA will get rid of those. So we have a bunch of those RNA chewing enzymes. RNA degradation that happens very readily. Okay, so we have a bunch of those RNA chewing enzymes. So for example, this RNA molecule, if it's exposed to this enzyme called RNase A, it can cut up these two nucleotides into pieces. That's a degradation we're talking about. So since there are RNA chewing enzymes everywhere, so it'll be hard to isolate and use RNA for our northern blot analysis, because before you even use those RNA molecules tend to be degraded with those enzymes. So we need to be careful when you deal with the RNA molecules. So these are suggestions. So you can prepare a separate set of glassware and maybe instruments, apparatus for your RNA-related work, like including northern blotting. And then once you have those separate set for your RNA-related work, those are set of glassware or whatever, the things that will touch RNA molecules, they will be treated with autoclaving. Okay, and then autoclaving actually is not enough to remove RNase enzymes, so they'll have to be baked at a higher temperature for a long time, like 300 degrees in Celsius for about 4 hours. And then maybe the RNase enzyme that can be inactivated. Another chemical that will help is called DEPC, it's diethyl pyrocarbonate. That's the full name. That tend to inhibit RNase enzymes because it's got unique structure. Okay, so it binds on the active site, it inhibits RNase enzymes. So you can coat up those glassware and whatever the apparatus that will be used for your RNA-related work with DEPC. Okay, then RNase enzymes will be inactivated. So these are kind of treatments that will prevent the degradation of your RNA molecules before you use them. Okay, those are important steps you need to do. And then once your glasswares and the things that you're going to use for RNA work are all treated with DEPC and then they were baked, then you can continue RNA purification. So you grow up the cells and try to isolate RNA molecules. So in isolating RNA molecule, the key is the speed. Before your RNA is isolated and degraded, you're going to do everything very quickly. Okay, and then you freeze them. Okay, and then normally strong denaturants can help. Okay, to break open the cells and isolate RNA, and then they can also denature RNase enzymes, so you're not going to have RNA chopped up during your isolation process. Okay, so the strong denaturant would help to inactivate the enzyme. That's one thing. And then phenylchloroform, isopropanol extractions are used. The same process that you use for DNA isolation and RNA isolation as well. And then RNA pellet is suspended with a DEPC treatment, or even the water you use should contain DEPC as well to inactivate the RNA chewing enzyme. And then RNA can be quantified using spec reading at 260. Remember that reference point. 260 equals 1 is about 33 microgram per mil of RNA. So you use that. Once you've purified enough cellular RNA molecules, now you can fish out mRNA molecules only by using what? By using a little column that contains poly T nucleotides. Then they will bind with your mRNA molecules that have poly A tails. So that's the additional step to fish out mRNA molecules and remove other RNA molecules that you don't want. Okay. Once that's done, then you can do your gel electrophoresis. So your mRNA molecules will be separated out based on their sizes using the electrophoresis, and then the gel will be denatured just like southern blot hybridization. Okay, electrophoresis and denaturation, and then but before that, one important step. Okay, so glyoxal or formaldehyde is used in the gel. Okay, so that's a kind of denaturant. Because your RNA is single-stranded, so you can make a bunch of those hairpin structures. To remove those hairpin from RNA molecules, you add these chemicals, formaldehyde and glyoxal, and your RNA single strand will be relaxed. Okay, so that treatment is needed for your RNA to be denatured, so you can it doesn't have any secondary structure, so you can bind with the probe. That is important. Okay, and then RNA can be visualized with ethyl bromide staining if you want to. Okay, and then the rest of the process are the same. transfer of the RNA onto the membrane using the capillary transfer process. So membrane is nitrocellulose, nylon. We already talked about that. In the southern blot hybridization method, it's the same method. Okay, and then